Our second speaker this afternoon is Sam McKee. Again, you've got his biography if you brought it with you, or you can look it up later. And Sam's going to speak in defense of human gene editing.
Yes, so if you've got rotten fruit or something to throw, just maybe keep it to the question time. Okay, right. I'm here to defend human gene editing, including germline editing, from a Christian perspective. Ambitious maybe, but hear me out for a second. Oh, yeah, I need this thing, don't I? There we go. Right, biology is in its biggest boom since the 1950s. I worry sometimes that our emphasis on discussing artificial intelligence has maybe given us too much of a blind spot on discussing and engaging with just how serious biology has advanced and exploded in the last 15 years. I think its recent developments have rivaled the 1950s in terms of how quickly biology is moving. We've got interweaving advances in cancer immunotherapy, which got the 2018 Nobel Prize, structural biology, which won the 2024 Nobel Prize, gene sequencing, and genetic engineering. The scale and speed of the advance far exceeds public knowledge and public discussion of it. It's transforming possibilities and practicalities, and it can't be put back in the box. In 2023, we saw the applications of gene editing technology for sickle cell disease that had made some breakthroughs in the US make their way over here. And also earlier this spring, we had gene editing breakthroughs in beta thalassemia make their way over to the UK as well. Cancer treatment will change. Antimicrobial resistance will change because of gene editing and what we can do with this new weaponry to combat those. Let's just quickly look at what it is, for those of you who don't know. CRISPR-Cas9, which the 2020 Nobel Prize was awarded for to Jennifer Doudna and Emmanuel Charpentier, it borrows from the bacterial immune system. CRISPR stands for clustered, regularly short interspaced palindromic repeats. We'll test you on that later. It scans the cell for foreign DNA, and then when it recognizes one of the spacers and thinks, wait, I know you, you're a viral invader, the Cas9 protein comes along and cleaves it, and that's the end of that. You can use a guide RNA to insert, and as long as it's got the right sequences at either side, you can basically use CRISPR-Cas9 like a word processor to cut, copy, and paste whatever you want into the genome. The possibilities of this are huge. The full genome is available, and we can now borrow from nature's toolkit. And it's a pretty big toolkit. Such unparalleled access raises massive ethical questions, especially when it comes to the germline. So why should we advance with human genome editing? Reason number one is because genomic medicine is the next revolution. It is the next great leap in medicine, is genomic medicine. One can now study genomic medicine as a postgraduate at Cambridge, and Cambridge isn't the only place. The next huge leap is in enhancing our medicine and overcoming the genetic challenges of pathogens, tumors, and the like. The dream, of course, the dream of medicine is personalized precision medicine, where you are treating the individual according to their own unique physiology, their own personal genetic makeup. Rather than using grenades and rockets, you're using sniper rifles. Rather than saying, well, this drug should work on people, you're saying this drug is tailored for you. And that's how we're going to treat you. Tailored towards the patient's individual unique physiology. We want to treat individuals as accurately as possible. Gene sequencing is now so cheap, so easy, and so quick. What cost over $3 billion and took 13 years with the Human Genome Project nearly a decade ago was done with a handheld Minion device in space on the International Space Station in a day by NASA astronaut Kate Rubins. You can now, in the field with a handheld device, sequence the genomes of organisms for less than £1,000 in just a few hours. So when you can sequence people's genomes and treat them according to their own makeup, You can do so much more, so much quicker, so much more accurately. And this has always been the goal, and now it's on our doorstep. Here's a big reason for you. It's going to get better with fewer issues. A lot of the fear and anxiety is around off-target effects. Well, in 2020 and 2021, along came base editing and prime editing to rival and complement CRISPR-Cas9. And what base and prime editing allow you to do is to make an individual change to an individual base, rather than using chunks or cassettes as you do with CRISPR. You also don't break the DNA backbone and make use of the human repair mechanisms. You're just changing a base individually, one at a time. And this is going to greatly reduce the possibilities of off-target effects. Once this uncertainty comes down and accuracy and experience increase, it's just going to get better and better with fewer issues. You know, in the run-up to this talk, I just did a quick jump on PubMed and looked at how many papers referencing CRISPR were published in the last week, and it was over 600 just for CRISPR alone. So the research output on this is enormous. But let's go to some theology for a moment here. The call to healing. Everyone I know who's got into genomic medicine or cancer genetics, it's the call to healing that's drawn them there. I love the verse in the New Testament in the Gospel of Luke that just says flippantly, and Jesus healed them all. And when we remember in the Great Commission at the end of Mark's Gospel, not Matthew's Gospel, our command is to go and heal the sick. Jesus was always healing people. Some genetic diseases can be vanquished. Some diseases that have plagued humanity forever could disappear, like smallpox, Tay-Sachs disease, Huntington's disease, sickle cell disease, could all theoretically be gone in a generation with this. Furthermore, as we borrow from nature, we can alleviate the effects of climate change and extreme environments for people who are living in more extreme environments. I'm doing a presentation at the British Interplanetary Society next month about the use of gene editing technology to help people survive in the harsh environment of Mars. And the knock-on effects of that for how you could live in places which are extracted by extreme environments. They are a pipe dream, but at least the technology there now is there to make it more possible. This will be the great equalizer also against tumors and antimicrobial resistance as we arm and tailor our drugs towards the individual's unique physiology, or perhaps to the tumor signature itself. Christians built all those early hospitals, and I think we can carry the way forward. I wrote a short piece on this, just a think piece for Science and Christian Belief. And this isn't the topic of my talk now, but this is something that I'm quite passionate about, which is Christians used to be the early futurists. Christians used to be the great futurists. When you look at the birth of the Royal Society, they were all Christians, and they were there at the front looking to harness science for the good of humanity. They were on a mission to use science to make everything better and improve the lot of the common man. We have largely ceded this ground to the humanists and to the secularists. When you think about who are the great futurist writers whose books you'd get in Waterstones, perhaps you'd have Lord Martin Rees or Neil deGrasse Tyson or Yuval Noah Harari or Steven Pinker, the people who are saying, look at technology and how it's going to make us better and are communicating that to the world, are all the humanists. And it used to be us. The Christian voices for the future now tend to be either, well, let's not think about a technological future because Christ is going to return in the next few years, and so we don't even have to worry about that, or the voices of extreme caution. And why I respect, and there is a place for the voice of caution from the Christians, and we should have that. I think we've ceded this ground far too much. Will it be the Christians who are saying no as we take the next giant leap in medicine? Because once upon a time, we were the voices at the front. And finally, it is inevitable. I mean, we can't put it back in the box, gene editing, where it's going and what's happening. And I was an undergraduate at Cambridge in 2019 when the He Jiankui affair happened, when he used CRISPR-Cas9 to edit the genomes of two unborn twins to confer on them a resistance to HIV. And our lectures for the day were cancelled so that we could have an in-class debate about the ethics of this. He Jiankui was put in prison. His lab was shut down for 3 years. And just like that, he's out and he's been given his lab back and told to just carry on with your research as if nothing has ever happened. It is inevitable. So what that means is Christians should be aware. Christians should be involved. Christians should be at the forefront of genomic medicine so that we can be a voice with the ethical mindset who are guiding it in good and healthy directions, asking the right questions, but also involved in the next great leap for medicine. Thank you very much. Only so much I can say in that time, but there you go.
I'm sure that will prompt some questions.
Maybe ask Dennis last.
And you have saved us a minute or two. So that's good.
How is this actually supposed to work? Somebody goes into the hospital with a tumor. So what do you actually do to that person?
Well, it depends on how you're treating them, but what this looks like, and there are ethical questions about people's data that come with this, but everyone has their genome sequenced, and then because it's quick and relatively inexpensive now, you will then treat, well, if it was cancer in particular, obviously you can sequence the tumor. You could arm your immunotherapy drugs a lot more individually. A lot of this is hypothetical. We're at the beginning here. But basically you have more information, much better information to work with in terms of how you treat them.
So you will design a drug that will treat that tumor?
Yeah, in theory.
Thanks, great talk, Sam. Enjoyed that. A lot of people might say, okay, so in fact, for the diseases you mentioned, we have pre-implantation diagnosis that achieves the same end. It weeds out lethal genes out of particular family histories and so on. What would you say to that?
Yeah, I think that the hope here is that some of those genetic diseases like Huntington's or Tay-Sachs could be wiped out. Now that would be germline editing. You'd be editing the faulty gene in people so that subsequent generations don't have that. I know it's strongly theoretical, but it's possible. Yeah, sorry, go.
Just come back with that. But pre-implantation diagnosis, you get the same end.
Yeah.
You're weeding out of the germline by preventing the birth of babies without carrying that particular mutant gene. So you could argue, I mean, some people do argue, you're achieving the same goal without necessarily going through the rather dodgy and problems of doing germline editing. So.
Yes, yeah, they are two routes to the same goal. I think the thing is the germline goal means people who have the genetic diseases, they could still have children, they could still get married, and rather than, okay, I can't have this pregnancy, or with genetic counseling with couples with Tay-Sachs disease, it's like, okay, we can't get married and have children because they will almost certainly have the faulty gene. Now you're achieving the same end, but maybe in a way where people can still do that. But again, I know it's theoretical. And yes, you're right, it's two ways to the same goal.
I read that every person has got 5 errors in their genes. So are we all going to get edited? And if so, will the NHS be overwhelmed?
No, I think that the hope is actually this could alleviate some financial burden on the NHS because using CRISPR is so cheap and genetic sequencing is so fast. Basically, if you're doing everything a lot more accurately and a lot more individually tailored, there should be less misses and more hits, and that's the hope anyway. But no, I'm not advocating for, obviously the slippery slopes here are designer babies and do you want to edit yourself to be smarter and stronger and all that sort of thing. I'm not advocating for being cavalier with the human genome, but I am advocating for a move forward, and particularly with the medical side of things. Let's use this new weapon to hit at some disease. All those errors you're talking about, a lot of that is what makes us who we are as individuals. So I'm not using this as in a eugenic sense to create the perfect man and the perfect woman, but rather kick diseases butt.
Last one.
Really good talk, Sam, thank you. So we know that some genetic diseases have an evolutionary advantage. For example, sickle cell anemia protects some people from malaria. By curing genetic diseases, are we not opening up a Pandora's box where we might become susceptible to other diseses? And how do we handle that? Do we just continuously keep
we just edit that, and then we edit that, and then we edit that, and then we edit... No, I'm kidding. Malaria is an interesting one because CRISPR is being put to use against malaria with gene drives, where populations of mosquitoes are being edited in a way to severely reduce the spread of malaria, which some of you will know well about. And there are ethical questions about that. Should we just edit populations of mosquitoes because we don't like them, but obviously they're important for the food chain? Yeah, and again, we shouldn't be cavalier on that. Yeah. Thank you.
So I think there's going to be some good scope there for getting our forum going for some hot debates on that subject. Shall we say thanks to Sam again?